my name is Krisha and I'm your medtech for today. I'll show you how we properly accept samples in the laboratory. I have just received a tissue sample and the request form right here. Tissue specimens received at the surgical pathology lab are accompanied by a request form containing patient information and history, as well as a description of the site of origin. The first step is to make sure that the specimen is in good condition and securely sealed, and we also have to make sure that it was prefixed to formalin. Examine the request form to ensure that the information on it conforms to the information on the specimen container. The name, age, and gender of the patient, the type of specimen collected, and the date and time of collection should all be written. After verifying, note down the serial number in the specimen container, as well as the request form. SP stands for surgical pathology, and 21 represents the final 2 digits of the year it was collected. 154 represents the year's specimen control number. Finally, write the serial number, name, date of birth, address, specimen, pathologist, date specimen received, and due date of the specimen in the logbook. And that's how samples are received in the laboratory. Thank you!
Making a paper boat would be advantageous if tissue cassettes were not accessible in order to proceed with tissue processing. The only item required is a piece of glossy paper, such as the kind seen in magazines. You trim the page and cut to the size of a 1/8 yellow sheet of paper. After that, you fold it 3 times lengthwise as well as 3 times crosswise. By now, we have 9 boxes, and we now fold the corner boxes and let the perpendicular lines meet. The 4 flaps will now be folded towards each other, and the resulting flaps will be folded outwards. And now, we have an improvised cassette for our specimen.
Fresh tissue examination is usually done for a rapid microscopic analysis of a specimen while surgery is taking place. Protoplasmic activities such as mitosis and phagocytosis may be observed when tissues are examined in its fresh state.
Methods of preparing fresh tissue mounts: teasing or dissociation, squashing or crushing, smearing, and frozen section.
We will now be performing the different techniques in smearing. Streaking. First step, pour a small amount of the sediments on one end of a clean glass slide. Second, using an applicator stick, streak the sediments in a straight or zigzag line throughout the remaining length of the slide. A relatively uniform distribution of the material should be obtained. Third, cover it with a cover slip. Fourth, label the slide. Spreading. First step is to transfer a small amount of the sediments on one end of a clean glass slide. Second, using an applicator stick, spread the sediments throughout the slide. Then with a circular motion, gently spread it into a moderately thick film using an applicator stick. Third, Label your slide.
Pull apart method. First, place a drop of the sediments at one end of a glass slide. Second, cover it with the end of another glass slide. Allow the sediments to spread evenly between the attached surfaces of the two slides. Third, with a single uninterrupted motion, pull the two slides apart in opposite directions. Cover the smear with a cover slip. Fourth, label the slides. Touch or impression smear. Imprint method. First, cut the block in half, exposing a fresher surface. Second, allow the freshly cut surface to come in contact with the surface of a clean glass slide. Third, apply gentle pressure, allowing the cells to be transferred directly to the slide. Fourth, cover the smear with a cover slip. And fifth, label the slide.
One of the many steps in preparing the tissue specimen is through teasing technique. So from our frozen block of tissue, we cut several small thin slices. After cutting it into thin slices, we then immerse the tissue specimen in the petri dish containing the saline solution. Then, using a dissecting needle, carefully tease the thin slices of tissues until they unfold and spread out. For the last step, carefully mount a teased slice on a slide and cover it with a cover slip. We also have squashing technique. To properly perform the technique, we will utilize the fourth step of the teasing procedure. Then, we will apply our methylene blue stain directly on the tissue slice on the glass slide. After applying the stain, cover the stained specimen with our cover slip and apply enough pressure to crush the specimen flat. Lastly, we will label our specimen. And that's it. That's the squashing technique.
Good day everyone, I am Maria Eloise M. Reconalia from BSMLS 2B and I am going to demonstrate to you the manual fixation in tissue processing. First, put the cut specimen in a tissue cassette. And since I don't have any tissue cassette, I used gauze pad and tied it using a string. Next, place the specimen in a container, ideally a beaker, but since I don't have any beaker, I resorted to a transparent glass. Next, fill the container with fixative. Next, cover the glass with foil. Lastly, label the container with its name and code. And that's it for our manual fixation.
Good day and welcome. My name is Arif Manulon and I'm going to demonstrate to you the decalcification process. So after fixation, if the tissue specimen is calcified such as the bone, teeth, or blood vessels with atherosclerotic plaque formation, they have to undergo decalcification first before proceeding to the next step, which is dehydration. This is because removing calcium would help in preserving the important microscopic elements of the tissue. So let's start first with the materials. First, we're going to be needing 500 ml beaker, 10% nitric acid, concentrated ammonium hydroxide, saturated ammonium oxalate, foil, litmus pH paper, pipette, and lastly, our tissue specimen. So for the procedure, first, we have to place the pieces of calcified tissue into the 500 ml beaker filled with 10% nitric acid. Second, we have to label our beaker with our group number and section. Make sure not to forget sealing the beaker using the foil. And now we have to present the container to the laboratory stockroom for storage until the process is completed. While in the stockroom, we would allow the tissue to decalcify for 12 to 24 hours. And after that, we now have to test the extent of decalcification. So to measure the extent of decalcification, we have 3 different methods. But for this experiment, we would only perform the physical or mechanical test. This test would require you to touch and bend the bone or any calcified tissue immersed in the decalcifying agent. So let's try to bend this bone specimen we have to measure the extent of decalcification. So as you can see, the distal part of this bone are bending. And so that's it for our decalcification process. We may now proceed to the next step of tissue processing, which is dehydration. Thank you very much.
Good day, I am Erica Calcatapang and I will show you the dehydration process in the Histopath lab. But first, let us not forget to wear our PPE, our gloves, our eye protection, mask, hairnet, and lab gown. What is dehydration? Dehydration is the process of removing extracellular and intracellular water from the tissue following fixation and decalcification and prior to wax impregnation. Remember that the recommended volume of dehydrating agent is 10 times the volume of the tissue being dehydrated. Never forget to immerse tissue in an increasing concentration of dehydrating agent because placing tissues directly to concentrated dehydrating agent will produce considerable shrinkage and hardening of tissue leading to distortion. Here are the materials needed for the dehydration process. First is the forceps, our fixed tissue, and our increasing grades of ethanol. Ethanol is the most commonly used dehydrating agent and is recommended for routine dehydration procedure. Here is the procedure for the dehydration process. Immerse the fixed or decalcified tissue in a container filled with 70% ethanol for 15 minutes. After that, immerse the tissue in 90% ethanol for another 15 minutes. After immersing it to the 90% ethanol, we now immerse it for 15 minutes in an absolute ethanol. After that, we immerse our tissue in 100% ethanol for another 15 minutes. After 15 minutes, we now immerse our tissue to the third container of 100% ethanol for 30 minutes. After 30 minutes, we still immerse the tissue in an absolute ethanol but on another container for 45 minutes. After 45 minutes, at this point, all but a tiny residue of tightly bound water should have been removed from the specimen. Now we drain our tissue and proceed to the next step, which is clearing. After the dehydration process, I will now show you the process of clearing in the histopath lab. So what is clearing? Clearing is a process done to remove alcohol from the tissue after dehydration. This is done to facilitate the penetration of the tissue by the embedding medium. The recommended volume of the clearing agent is 10 times the tissue volume. Here are the materials needed for the clearing process. First is the forceps, our dehydrated tissue, and 3 containers with the most common clearing agent, which is saline. The clearing procedure starts with immersing the dehydrated tissue in the first container of saline for 20 minutes. After that, we will still immerse the tissue in the second container of saline for another 20 minutes. On the third container of saline for 45 minutes. After 45 minutes, we remove our tissue samples from the saline and place them on paper towels to drain them properly. After draining, we wrap in foil and store samples for the next histopathologic technique. And that ends the clearing process in the histopath lab. Thank you.